when we consider the plasma membrane of your cells, every single cell in your body is surrounded in a plasma membrane, so it must have a very specific structure and function. We have talked about the structure before. We said that it's made up of phospholipids, it's full of proteins, it has carbohydrates associated with it. There's a lot going on with this membrane. So what is its job really? Well, it separates the cell from the outside environment, and outside here means the inside of your body typically, right? So outside of the cell environment. Regulation, how do things move in and out? Communication, how do cells talk to one another? So we've talked about a lot of this already. I just want to put it in one place for you as far as thinking about what that means for the membrane itself. So it's a bilayer of phospholipids. It has proteins embedded and attached. It's what we consider a fluid mosaic. And it's fluid because of cholesterol and lipids. And then we have the combo molecules that we talked about before. So these are things like glycoproteins. So all of these things are going to work together to do those three functions that we mentioned previously of barriers, regulation, and communication. We know that phospholipids are made from unsaturated fatty acids. When we talked about this before, that meant that there were kinks in the hydrocarbon chains of the fatty acid. And that means that they do not pack together and they stay liquid at room temp. And they're going to be dependent on interacting with cholesterol. So cholesterol will actually fill in at some of those spaces that are left open to help to stabilize the membrane. Now this is good cholesterol. This is HDL. So this is the cholesterol that we need to help to stabilize our membranes both at increased and decreased temperatures. So it's going to work in both cases. This is kind of a mess, right? So you see the phospholipid bilayer. This is two cells, cell number 1 at the top, number 2 at the bottom, and then this space in between. So blue is always outside of the cell. And this kind of tan is inside cell. The purple, in this case, are proteins, enzymes, all kinds of things. They show you cholesterol here that's fitting in between to help keep that membrane fluid. We're going to attach cytoskeleton, all these things that we've already talked about, to make these cells work and function as they are supposed to. So like we said, kind of its major job of our proteins that we've looked at before is to maintain cell shape, and that's primarily by cytoskeleton connections. Having receptors in place, so these are going to be communication and transport and so on. And then enzymes, so to actually do some sort of job that's going to help the cell itself, and in particular, their role in metabolic pathways. We also look at glycoproteins being cell-to-cell recognition, intercellular junction, so how do you hold cells together, and then this last one is where we're going to spend most of our focus. This idea of selective permeability. It means that you will let some things in, but some things are blocked. And we'll look at what the difference is. This little bubble that I'm showing you here is actually water on the inside and phospholipids on the outside. So if you put a whole bunch of phospholipids into a beaker of water, it will automatically make these kinds of things. Why? The fatty acid tails are trying to hide from the water. They're hydrophobic, right? So they fold to the inside. The hydrophilic heads go to the outside and to the inside of the cell itself, or in this case, water droplet. And we get this membrane that essentially spontaneously forms in an aqueous environment. So this water kind of thing going on with a membrane in the middle with our hydrophobic tails and our hydrophilic heads. Looking a little more close up, we've talked about enzymes folding before or proteins folding before, and how the hydrophilic parts of the protein will want to be in contact with the water, and the hydrophobic areas will want to be in contact with those fatty acid tails in the middle. So the three-dimensional shape of the protein and how it folds with amino acids and hydrophobic versus hydrophilic, critical to the proper functioning of these enzymes and what they're going to actually do for our cell.